All right, so welcome to our another Chai and Wine. We are at a new venue today, HBCAC. This is an unusual venue, and today we are going to discuss the chemistry Nobel, which was announced just a couple of months ago, or is it? It's not even 2 months, right? And to tell us about that, we have two research scholars from TIFR, Mitradeep and Prithvish. They work in the group of computational chemistry with Professor Ravi, and all yours. Yeah. So let me just take the first round and then yeah. So they are going to take rounds to explain what. Yeah. So the presentation is shared. Or it's on the pen drive. Start the screen share. Yeah. Okay. Okay. And you are also in the views. Great. No, you have to stand here. Yeah. Where literally you stands. Okay. Share. Let's start the screen share. Start here. Don't screen share. Yeah. No. From there. Yeah. And that's this one. Yeah, good. So I'm talking to a scientist here, possibly. Yeah. Yes or no? So you are, I met few of them in the starting. So you are a BSc student from bio background, and you are? Okay, you are from BSc, and you are a BSc in microbiology. Okay. No chemistry. Or biotech, microbiology, and yeah, physics. Yeah, physics. Right. And two chemistry people going to talk about which deals with biochemistry. So first of all, why are we here anyway? What makes this anyway interesting? Oh, restricting the screen. Now it's okay. So आप लोग क्यों आए हो यहाँ पे? क्या ऐसा बड़ा न्यूज़ था जिसकी वजह से आए हो? मतलब is it because it's a science talk or is it because it's a Nobel talk? अरे वाह, मतलब नोबेल प्राइज वो मटेरियलिज्म में बिलीव ही नहीं करते थे. अच्छा, नोबेल प्राइज मिलता है तो क्या इतना लाइक व्हाई इज इट अ बिग न्यूज़? व्हाई इज नोबेल प्राइज सो इम्पोर्टेंट? एनीवन? नो, नथिंग. इज इट द मनी? हाँ, रिकॉग्निशन. हाँ, मतलब इज्जत. एनीवे, तो this is like a typical banner of our what basically happened because of which there was a Nobel Prize recently, right? So all our biologists, everyone is familiar with proteins. Okay, everyone is familiar with proteins. Is it audible? Yeah. So proteins are the basic we will understand, but what basically this time the Nobel Prize was dedicated to was this protein building, and based on protein, whatever the structure is, this has become like a Lego building exercise right now to humankind, right? I will explain how. So just to summarize it, you all know that this is made up of amino acid sequence. And this is the structure. Of course, those two are related. You probably have heard about it. Now, this time Nobel Prize was dedicated to these people. Okay? So 50% of it was dedicated to these two people and 50% to this particular person. So what did these two people do? They basically revolutionized the field of protein folding by designing something which can predict the structure of a protein if you know the amino acid sequence.
So that problem is called protein folding, right? And what did this guy do? So if you want a particular structure, you can customize your amino acid sequence in a particular way so that you can get that particular structure. Okay. So when I say protein, since you are a biologist, you know more. But generally, to the raw organs, something like this comes to the picture. Some guy working out. Okay. There are all these whey protein supplements that people consume. Also, it's present in this. Present in this. People are non-vegetarian or vegetarian. Both. For vegetarians also, I have prepared something. So, anyway, why do people consume these things? First of all, to understand that, first of all, we need to know about what proteins are. So, proteins are what makes us, right? I want you to give you an exercise like what are the particular organs in our body which works without proteins? Is there any organ? Name one organ which works without a protein, which is not made of a protein. Let's just name one.
Is there any?
Yeah, I'm asking biologists. Like this is even not even my area of expertise. Right? I'm asking them. Like, think. Forget human. Like, go to your entire biological gene pool, whatever it is, whatever species are there. Try to think of something that exists in the earth where there is one particular organ or organelle which is not made up of proteins. Now, I don't know if someone of you are thinking about membranes. What are membranes made of? Phospholipid. So there is no protein there. How do proteins exist in the membrane? Like transporters, thing like that. Is there any other place where protein is not existent, which you know as a life form? I can't think of any basically. That's why I gave you this exercise. Think of something. And in Earth, basically all life forms are carbon based, right? So any kind of carbon based compound, you study it in 11, 12 as organic chemistry. Now, in organic chemistry, of course, all carbon compounds are not equally significant as an organic chemistry molecule. So proteins hold a special place. Why? Protein is the basic fundamental structural unit, like it is the structural building block of everything. And why is that the case? Because proteins have this interesting structure. So it is made up of these 20 amino acids. Popularly, this is the most known amino acid. Okay? So they form like these chains. Okay? Now, why these chains are so important? Whichever can form long chains is capable of forming long molecules. Now, okay, let's go back to 11, 12. Why was carbon so important? Like, what was that one property which made carbon so special among all the elements in the periodic table? Tetravalency. Is it और भी टेट्रावेलेंट है लाइक सिलिकॉन है या कार्बन के ग्रुप का कोई भी उठा लो सारे के सारे टेट्रावेलेंट है क्या दिक्कत मतलब व्हाई इज कार्बन सो स्पेशल यस कैटिनेशन व्हाट वाज दैट कैटिनेशन yes ability to form long chains now what happens is that this ability to form long chains is because of the very strong C single bond C. This is very strong, right? And also carbon can bond with other. This has to do with, of course, it's a second period element, so it's a very small element, right? So I will just give a brief like idea. In the periodic table, you remember a few elements, right? So the first period element are the smallest ones. You can kind of guess. Okay. So hydrogen, helium. Helium's problem is that it can't bind with anything because it's an inert gas, right? Hydrogen can bind, but it can bind with one. Now you come to the second period, right? So if you remember 11, 12 periodic table chapter, what have you learned? that as we go from left to right in a period, the size of the atoms decreases, right? Because more number of electrons are added to the valence shell, and there is this increasing nuclear charge, which is causing a greater attraction in the valence shell to the nucleus, which makes the size smaller, right? So from left to right, the size is going to decrease. So you hydrogen helium, then you go from lithium, beryllium, boron, till carbon it decreases, right? Now why at carbon it stops? Because beyond carbon there comes nitrogen, it comes oxygen. If you look at any of those nitrogen, oxygen, and fluorine compounds, you will see that there are lone pairs exist. So they do pose a repulsion, and which causes the bond length to extend. That's why it makes carbon. Carbon is a very unique element, which is very capable of forming this very strong bonds with itself, right?
Take some online.
Huh?
Can we take one online?
Yeah.
Why is it that there are only 20 amino acids?
So of course there are possibilities that other amino acids can form, but these are found in most abundant because whatever the natural machinery is there, it helps catalyze to form this particular amino acid possibly. And also this is one thing, if you are familiar with chirality, amino acids exist in the L form. So something intrinsic in natural machinery is there, which causes this particular amino acid to exist in a particular chiral conformation. Right. Anyway, so I explained that C single bond C is very strong, right? Now why? So amino acids have amine group and acid group, right? So there is this nitrogen involved there. Fat and these don't have nitrogen that much, right? Nitrogens are explicitly mostly present in our protein. What makes this nitrogen and this acid, amino acid, why is it so important? The reason is because of this. Sorry. So you see this existence of this acid part and this amine part, this poses the possibility that there can be a release of water and these two can bind together in a fantastic way. So this particular bond is called a peptide bond, right? Now, due to the existence of this peptide bond, these 20 building blocks of proteins, they kind of come together and form these long chains. And now the question arises: there is a long chain of these amino acids. Now, how do they fold? Like, what kind of shape does that chain acquire? So generally, you all probably are familiar with if you are from biology or chemistry background. So there are basically four kinds of structure as shown to you. One is primary structure. What is primary structure? It basically contains the information of what is the sequence of amino acids that form that particular protein. What is the secondary structure? Now we come into exact 3D structures, right? In a functional way. So how these particular amino acids are located in 3D space, forming small regions of proteins which are kind of a part of a same structure. For example, this represents a helical structure, right? It's like a spring. This is like a sheet. So you are probably familiar with these are called beta sheets, and these are called alpha helix. There are other kinds of helixes as well you are probably familiar with. And now there is this tertiary structure. Now, how these secondary structural regions they fold among themselves? That particular structure is called the tertiary structure. Now, I have not mentioned something. The next one is the quaternary structure. Now, how one protein is kind of interacting with another protein that falls under the domain of quaternary structure. Anyway, so of course, proteins do these are very broadly classified. Like you can also find other names for whatever functions they carry. Sorry. So of course, these properties, whatever it is, if it's acting as a transporter, or if it's so transporters are like kinesin, you probably have heard about it, right? Then chemical reactions, there are certain systems called kinases which catalyze a certain chemical reaction, right? So proteins are responsible for all these things that happen within a cell or even more. Now let's talk about the godfather of protein. Who is this person? Have you heard about it? Sir, I'm sincere. Biologists are not, you are not familiar with this person. So this person was also awarded Nobel Prize in 1970. Now why? He did a particular set of experiments. One of them was very popular. So this is a ribonuclease, okay? So this is a ribonuclease protein. So what he did was he added these two chemicals to it. One of them is beta mercaptoethanol, and one of them is urea. So beta mercaptoethanol is known to break something called as the disulfide bonds. So suppose there are two regions of chains which are bound by some sulfur bond. So beta mercaptoethanol is known to break those bonds, and this is known to break other weak interactions from the group. So what it does is that the combined action of these two basically unfolds this protein, this folded protein, which is appearing as a globular or blob-like structure. It's now basically unfolded into a chain. Now, what is the marker for that? That the folded state has a particular function. Let's not go into that, what function it does. And the unfolded protein, it loses its function. So, of course, there is one important message that whatever function a protein performs is dependent on its structure. Right? And it was also found out that since after removing these reagents, it is folding back to its native state. That means there is some information within the sequence of this protein which is causing it to fold in a similar conformation. You understand the word conformation? So 3D structure basically. Right. So this was Anderson's dogma. The native structure of a protein is determined only by the protein's amino acid sequence. Now, how? Right. So now the question is that if protein structures are so important, it is important for the scientific community to somehow get the protein structure. Right. Now, what are the routes? The experiment. These are probably how your biology or chemistry labs look like. What are the experimental techniques? So one of the big techniques is NMR spectroscopy. Another one is X-ray crystallography, and another one is cryo-electron. Something is in the way. Remove this. Oh, sorry. They are related. So yeah, cryo-electron microscope. Okay. So generally, these X-ray and cryo-electron, these are generally used to find out the structure of bigger proteins. Okay. And NMR is generally done for smaller protein systems, and it also has some dynamics information. Anyway, this is sorry, yeah, there is a problem. Sorry, guys. Yeah, the pointer is broken. Should I do it from here? Yeah. Yeah. Okay. So now you will do experiments. You're probably have you joined any lab in your BSc or you have? Yes or no? Yes. So there are so many equipments, right? So do you think lab is just like playing with this equipment? This equipment costs, right? A lot of money. So this equipment costs. So I just Googled these things. Okay, these numbers are from Google. You can also do the same. This NMR spectrometer, it generally costs between $35,000 to $17.8 million. Now you can do this homework. You can convert it into rupees based on the current. Now, X-ray crystallography is $30,000 to $45,000. $5 million to $6 million for special cryo-electron. Because cryo-electron, I don't know if you've heard about it or not. You freeze that particular protein and then look it through an electron microscope, right? So, how many of you can earn this kind of money in, I don't know, maybe 2 years in India? You can't think. Once it touches this M, then it becomes a big problem, right? Okay. We can maybe if you go in US probably and you can do a good job, you can probably save this much.
And only Adani can afford this.
Yeah, like it's not for middle class people. Right? So this is this problem. Another problem: how much time is required. So this looks like okay if you have equipment, like you just take the product. So there are a lot of things going on, right? You need to look for protocols to understand how to isolate that protein, how to set up that experiment to look at that structure, right? So the time required. So generally, I found this one. So how it started. You're familiar with this lady? Every night in my dreams, I see you. Anyway, my heart is going on and on. So yeah, how it started, how it's going. So you will one day you will join PhD and you will understand the deeper meaning of this. But anyway, so how to know fruit? So anyway, these experimental routes, yeah, they are first of all money demanding and it's also time demanding. Now we have computers. So generally computer-based techniques are called in silico, right? Where we use these setups. So this is a small laptop and this is a combination of many CPUs. Generally we call it servers or clusters or supercomputers. So generally these kind of equipments cost from $300 to 0.2 million. Now, most protein structures, as I will explain, it can be done using laptop and a slightly powerful equipment. So it won't take you that kind of, it doesn't start from the regime of thousands of dollars. Start from hundreds of dollars. Now, so the time required is however long you take to make this, or you take to make a biryani. Okay. So let's talk about this big thing. You have heard about it, right? AlphaFold. So AlphaFold 2, what's the big deal? Right? So you may not know this, but there's a global competition known as CAS, Critical Assessment of Protein Structure Prediction. Since I told you, since he emphasized the fact, okay, protein folding is an important exercise. If you want to know the structure, need to know, if you want to know the function, you need to know the structure. So since then, this global competition is being held where research groups from many places they participate. And they give their computer-based models, computer-based structures to see how good it is against experiment. So they have this pre-experimentally solved structures and they provide their in silico predicted structures and they see how good they are matching. So in this competition, this group called DeepMind was the winner. This DeepMind is in collaboration with Google and they won this competition. And the good thing about this, by 2020, right? So this violet one, since 2020, the violet one is all the experimentally resolved structures. Okay. All experimental structures. And by I think 2018 or 19, this was the only AlphaFold 1 based folder set. So already it surpassed the number of structures determined by experiment. Because as I mentioned, the computer-based algorithms are much faster. By 2020, it has almost 200 million plus structures folded. Now, whatever it benchmarked it against, what I mean is that when there was this CASP competitions, AlphaFold 2 was found, like it was known to give almost 99% or something, ridiculously high number of high amount of accuracy while solving the problem. It was almost considered, okay, if you have a single protein floating in this cytosol or something, whatever structure it is, AlphaFold will give the best prediction. So it's not just the accuracy of the structure that is of importance. You have to understand the speed it has reached, right? But again, what is so special about AlphaFold? Machine learning. So from here on, I am introducing my colleague Mitradeep. He is going to walk you through what machine learning is and why it is so amazing. So Mitradeep, so for you.
Thank you, Pritish,
Yeah.
for introducing the first part. So, how many of you guys have heard about ChatGPT? How many of you guys use ChatGPT to regularly do your homework? Oh, come on. And how many of you guys find clever?
Don't give it wrong.
Yeah. And how many of you guys find clever ways to hide the ChatGPT homework from being detected by your teacher that is done through AI? Only two people are okay. Let me put it this way: only two people are honest here. Oh,
They don't give homework.
you do not get homework?
Others don't give homework.
Oh,
Ask them if they use it for writing mails.
writing means doing assignments. Chalo, homework nahi, assignments. Okay. Assignments is glorified homework.
Texting girlfriends. Or boyfriends.
Okay, I think
Let's leave it.
let's leave that. Okay. So what is machine learning? Coming to the formal definition, the boring one. Machine learning is a branch of artificial intelligence and computer science that focuses on using data and algorithms to enable artificial intelligence to imitate the way humans learn, gradually improving its accuracy. Most boring. Question is, how do machines learn? How does a human learn? Read. How does a machine learn? Read. Or is it? What do you guys think? Okay. So it's all about pattern recognition. So for example, this is one of the standard set of letters used for training machine learning for handwriting recognition. So if I ask you what is this letter at the bottom left? 6. What is this letter? The top row fourth column? What is the bottom right one? But you see this 6, and this 6 structure is different. Still you can understand that 6. A machine can also do. So this is one of the classic data sets used for let's say a homework exercise or assignment in machine learning. So it can do pattern recognition. It can also do image recognition and generation. And for your information, these two images both are AI generated. So how does machine learning help in protein folding? So this is actually the working algorithm of AlphaFold. Credits to the AlphaFold paper, they wrote it in such a beautiful way. So let's say you have a protein which is helpful in, let's say, for muscle movement. You want to, you know, a sequence, you want to know its structure. What do you do? Simple thing. You look for what if we have structure for proteins which help in muscle movement in eggs, in chicken, in fish, in monkeys, whatever it is. Right. Also, so this is the first part, which is called multi-sequence alignment, where you look for other organisms having similar proteins and you look if they have a similar structure. The second thing is, if you have a sequence, you look, okay, fine, can I get this sequence in different proteins, which may be related, may not be related. And then you feed this information into the machine learning algorithm, which then uses it to generate structures. So this is not like a one pass. You have to iterate it a couple of times. And finally, you get a structure which is colored on the basis of how confident the algorithm is about a region. So whatever you see here in blue is the high confidence region. Whatever you see here in orange or red is the low confidence region. So this is how AlphaFold works. And as my colleague Prithvish has explained, it has expanded the database of available protein structures by, I would say, how much it is? 100 times?
More than way more than.
Way more than 100 times.
If you looked at that circus, right? That was published by DeepMind.
So can we fold a protein right now here in this room? So take out the smartphones. If you have network, well and good. Otherwise, you can use this Wi-Fi. For the online audience, you can open any browser in your computer or on your smartphone and go to this website, alphafoldserver.com. So, huh? Can I do screen share on this? What I've already. And so protein uses 20 amino acids. And these are the letters of English alphabet. So these are the 20 amino acids which you can use. Rather, the interesting one to remember is do not use these 6 alphabets. B, J, O, U, X, and Z. They do not correspond to any amino acids. Rest you can use. So for a slightly advanced audience, the ones in the red are the hydrophobic ones. The ones in the green are the acidic ones. Yeah, the ones in the blue are the basic ones, and the ones in the yellow are the other amino acids, mostly hydrophilic ones. So, did you get to that website?
Yeah, I am sharing.
Sure.
Okay.
So I'll just stop sharing from here. How do I? Oh, red. This red button. Okay. Okay. Yeah. So what we can do here in this protein sequence, let's put up any random sequence of, let's say 20, 30, 50 amino acids, whatever you wish. Okay.
How about what we do? Let's ask them one alphabet of an amino acid. I will just put them down.
Right.
Yeah.
Okay. So I'll just put up the PowerPoint screen for the audience here in for your reference. So one alphabet. Let's start. Huh?
From the last bench, maybe.
Okay, from the last bench.
Yeah. Give me one alphabet.
One alphabet in this table. I,
Full.
W, K, F, R, W, M, N, A, S, D. One more. A, Y, L, S, I, M, K, Q, I, C, V. So we have roughly 20 amino acids.
Right.
Okay.
Yeah, it's 22.
22 amino acids. So we have 11 audience here, 2 alphabet from each one, 22 amino acids.
Yeah.
And yeah, I'll just
show them that screen.
sure. Sorry, zoom screen then. So this is the sequence you generated. None of us know what this protein is. None of us have seen anything close. Let's see how it forms.
So I'm just submitting this job.
Right. And it takes quite a few seconds. Maybe you can scroll down, it will show the list.
You
Yes.
see? Yeah.
So it's still running.
Till the time you can continue.
Yeah, you can put up your own sequence on your smartphones as well. I'll bring up that PowerPoint slide once again to help them look up which alphabets to avoid.
By the way, this job is done.
Okay. So we have a folding for the sequence that you have given, and see it's a few minutes ka thing. So let's click on that. And
Today,
yes. And here you can see, wow, you guys made an alpha helix. Pretty unusual. And you can see this is like full blue. That means AlphaFold is predicting that it has a very high confidence. And if you synthesize this sequence in a peptide synthesizer or inside your cell, most probably it's going to have an alpha helix sequence. So
They can try that in their college lab.
yes, you can. It's just like an online game. So anybody of you got some interesting sequence? Some interesting structure? Not with this sequence. You guys must have gotten your own sequences, right? So if you scroll down, you click on the job name, you will have the structure. So whosoever has got the slightly complicated structure, something interesting, I'll show on the camera to the online audience as well. So you have gotten something interesting? Okay, let's see. Okay. Okay, so what's your name? Anushka. So Anushka has got a broken sequence. Okay, this is interesting. For the online audience, can you please stop screen sharing?
Yeah.
So for the online audience, this is how it looks. Is it visible?
It is delayed. Link sprint delay.
So she has got a broken kind of sequence. Anybody else got something interesting?
Yeah, it's visible.
Maybe somebody else can show what the folded protein looks like. Alpha helix? Huh?
Still loading.
Still loading?
Loading.
Okay.
Okay, what we'll do is we'll go back to this after the.
Sure.
Yeah, and you
Yeah.
can go ahead with the.
Sure. So maybe
So
we
let that job run. What we'll do is after the session, we'll have a small tea break. And then of course, the speakers will be at your mercy. You can ask them whatever you want.
okay. So coming back to the discussion. So we just folded a protein sequence, which we made up. Now let's talk about the other question. Suppose you want a structure. Can you make a protein sequence accordingly? It's a slightly more difficult job, or rather it's not like a wow moment. So I'm going to present a timeline. So the first synthetic protein was made in 1997. How many of you were born on or before that year? No.
You're making us feel older.
Yeah, I am old. I am 97 born. My other speaker is 94 born.
why are you doing that?
Don't ask Mike.
Okay, let's spare the organisms. So this is the first de novo protein. It's made from an idea of a zinc finger. So zinc finger is a protein which binds to zinc in your body, and it kind of traps the zinc and help in various bodily functions. Then comes our Nobel laureate David Baker in the picture. Although there have been other people who have been designing proteins, but from now I'll be discussing what David Baker said. So in 2003, he came up with a protein which folds in a new way, and this fold doesn't correspond to any of the naturally occurring proteins. So it's called top 7, and it has 7 secondary structure components. So you can see the 5 beta sheets at the back, and then on the front is 2 alpha helices. So it's like a sheet with 5 beta sheets, and then on top there are 2 alpha helices on top. Then in 2008, he designed first de novo enzyme, which performs organic this thing. I think it's chems elimination. So there are 2 enzymes. One is chems elimination, and the other is some kind of alcohol to aldehyde or ketone. And both of those didn't have any natural enzyme. So he designed an enzyme that does that. In 2016, he designed a protein-based nanomaterial, and the list goes on and on and on. So some of the other things that David Baker has designed is he designed a fentanyl binding protein in 2017. Do you know what is fentanyl? Anybody has any clue? Come on, you are biologists. You must know this. So, okay. So let me tell you, it's a synthetic opioid, and it's banned in almost all countries in the world due to its severe health effects. So this protein is supposed to detect fentanyl in the blood or whatever solution was in it. Then there is a molecular rotor he designed in the same year. Then he designed a protein nanoparticle complex, which imitates that of an influenza virus. And do you know why this is important? Any guess? Random guess. Think creatively. Why do you want a nanoparticle with proteins that represent influenza virus? Yes, for developing vaccines. This year he designed a shape-changing protein, which changes the shape based on physiological conditions. And no, I'm not talking about unfolding here. And this is one of my favorite. He designed a protein that binds to something called a spike protein in COVID. So COVID has this first line of attack, which is the spike protein. So it's a completely novel protein, and it's a much better binder than whatever your natural binder to the spike protein is in the human body, which is ACE2. Anyway, lot of scientific terms. So the problem of protein sequence to structure is done. Structure to sequence almost done. What next? So as you might know, protein is not a fixed structure. They're mobile objects, and they can stay in different states while they're performing the function. It's essential for us to know what are the states, how they interconvert, what is the relative populations, what are the pathways of function. And that is where we come in. So, yeah. So my research involves studying pathways and the populations for this thing. So I am developing some statistics based and statistical mechanics based tools for that. So yes, we are using physics to do biology. So for example, this is a protein that is in the HIV protease, and it has this very distinctive close to open motion, which helps it do its function. So you can get out what are the pathways of it, what are the free energy surface for that. And my colleague Prithvish.
Yeah, hi again, guys. I'm back. So right now, I will also talk about a bit of Mitradeep's contributions in our lab. He has been developing so many tools for us to use in while studying actual biological entities. Okay, like one I am using now. I will give credits in paper maybe, not. But anyway, so right now I'm looking not at a protein. I'm looking at a protein complex. So what is the difference between a protein and a protein complex? Protein is a isolated single chain, single molecule. When such big molecules form a conglomerate, then we call it a protein complex. So I am studying this particular protein complex, which is named mTOR complex 1. As biologists, you probably have heard about it, this name, mTOR, right? Or maybe not. So it's basically a kinase system which doesn't work alone. Basically, so these blue ones are the kinase system of interest. It works while being binded to all the other proteins that you can see. These two structures are solved using cryo-EM, which has a lot of flaws. It has unresolved regions. Okay. And also it has a poor resolution, the cryo-EM structure. It has a resolution ranging from 2.75 angstrom to 7 angstrom. So right now I'm working at a method where I use AlphaFold 3, by the way. We haven't mentioned this. AlphaFold 2 has taken another step to predict structure of not only proteins, protein complexes. So I'm using AlphaFold 3 and I'm trying to improve the model of AlphaFold 3 because it is still not perfect because there is some particular phenomena which we study in chemistry known as allosteric effects. Now, what is the allosteric effect? Due to one protein binding to another protein, there is some chemical reactivity change somewhere else in that second protein. Now, AlphaFold 3 can predict the interfaces, right? With predicting two protein structure, it can predict the interface very well, but it cannot predict its action at that distance where the chemical reaction is occurring. So I am trying to use the cryo-EM data to improve the model provided by AlphaFold 3. That's what my project is. So I think I will let Mitradeep conclude this thing. So yeah, Mitradeep.
Yeah. So let's come to the end with an interesting story. The date was 23rd February 2024. The place was Taj Hotel, Mumbai. And one of the Nobel laureates, he was not a Nobel laureate then, came to visit us at TIFR. And we had a discussion with him in this Taj. So we had a lot of discussions with him. Who is this person? John Jim. Right. And the person you see beside him is Professor Sudipta Maiti. He's a well-known biophysicist and a biochemist and was then the head of the chemistry department. We had a lot of photographic sessions with him. These are various faculty members of our department. And we also had chai with him. So he is actually our guide, Professor Ravindra Venkatram. So this was the first half. And in the second half.
First chai and wine.
Yeah. Yes.
They attended this talk.
So you attended this lecture, right?
Oh, sorry.
So he told us a lot about it. And interestingly, in both the lecture and this session, both of us were there. So conclusion of the story, you want a Nobel Prize? Give us a party in Taj, both of us. Okay.
All right.
Jokes apart. So with this, we conclude the session. And these are some of the references we have used to get the photos and stuff. And thank you all. And these are our, for the online audience, these are our email IDs. Please feel free to write to us in case you have any questions, comments, queries, and so on. Thank you.
So thank you. Thank you, Mr. Deep and Prithvish, for a very interesting talk. And
So what we do is we close the online session. This session is also basically it's a hybrid mode. So we'll take a break. We'll come back to this room and then you can ask them whatever you wish. Feel free. So we'll close the online. Okay.